Welcome to The Explainer. What if I told you we could design a life-saving vaccine before anyone even touches a test tube? Well, today, we're diving into exactly that. A groundbreaking new frontier where we build our defenses against deadly viruses entirely inside a computer.
So, our story kicks off with a very real and very dangerous threat. It's an emerging pathogen called the Chandipura virus, and it's been the cause of several serious outbreaks in India.
And when I say dangerous, I mean it. In one major outbreak among children, the case fatality rate was a staggering 78%. And what makes it even scarier is that right now, there's no vaccine or treatment available to stop it.
This really brings up a critical question for public health in our modern world. When a new viral threat like this pops up, how in the world can we develop our defenses much faster?
All right, let's get into it. First things first, we need to properly meet the villain of our story, the Chandipura virus itself.
So this virus is spread by sandflies, and it causes this severe inflammation in the brain, mostly affecting children. The illness moves with terrifying speed. We're talking high fever and convulsions that can lead to a coma or worse in just 48 hours. With no treatment out there, the need for a vaccine is absolutely urgent.
Faced with this pretty dire situation, researchers turned to a really revolutionary strategy, designing a vaccine completely in silico, which is just a fancy way of saying entirely on a computer. This whole cutting-edge field is called immuno-informatics. It's all about using some serious computational power to study the immune system and predict how our bodies are going to react to a disease. This lets scientists design and test treatments virtually before ever stepping into a wet lab. And here's why this is such a big deal. It's all about speed and efficiency. You see, traditional vaccine development can take decades and cost a fortune, with a really high risk that it'll fail late in the game. But this digital approach, it can pinpoint the most promising ideas in just a matter of months, saving an incredible amount of time and money.
So, how did they actually build this thing? Let's walk through the exact digital blueprint the researchers followed to construct their vaccine candidate completely from scratch.
Okay, now I know this looks a little complicated, but don't get bogged down in the details. What you're really looking at is the entire process from start to finish. Just think of it like a digital assembly line for a vaccine, taking you all the way from analyzing the virus to having a final product ready for virtual testing.
It really boils down to 3 key steps. First, they digitally took apart the virus's 4 main proteins. Then, they used some smart algorithms to predict which tiny little pieces of those proteins, they're called epitopes, would be the most obvious to our immune system. And finally, they digitally stitched the best of those pieces together to create a single vaccine molecule.
A really great way to think about these epitopes is to imagine them as most wanted posters for the virus. The whole idea is to show the immune system the most distinct features of the bad guy, so it knows exactly what to look for and what to attack.
And here it is. This is the final digital product. This 3D model is the potential vaccine, a molecule built from all those most wanted posters, which the researchers gave the name MEC-CHPV.
But hey, a fancy 3D model is one thing. The real question is, would it actually work? To find out, the researchers put their digital creation through what was basically a completely virtual clinical trial. So, their first test was a technique called molecular docking. This is basically a simulation that predicts how well the vaccine protein will bind or dock with the key receptors on our own immune cells. Think of it like testing if a brand new key fits into a lock. In this picture, the vaccine is the red key and the immune receptors are the blue locks. And as the simulation showed, it was a perfect fit, a really strong, stable connection. Okay, so with that successful docking confirmed, the key fits the lock, they moved on to the next big step, a full-scale simulation of the entire immune response just to see how the body would react over time.
And the results? They were incredibly promising. This chart from the simulation shows a classic, powerful immune response. You can literally see the spikes in antibodies after that first virtual dose, and then even stronger responses after the second and third simulated booster shots. So, to put it simply, the virtual trial was a huge success. The simulation predicted everything you'd want to see: strong antibody production, the release of critical virus-fighting signals, and ultimately, a well-trained immune system that was ready to clear the threat.
So what does all this mean? What does a successful virtual experiment like this actually tell us about the future of medicine and how we fight off these terrible diseases?
Well, the scientists who did this study put it perfectly. They said this approach provides a cost-effective and time-saving way to design a vaccine candidate. It's a true game changer, especially for that first really difficult phase of vaccine discovery.
Now, it is really important to remember that this is still a vaccine candidate. The computer has done its job. It's given us a fantastic blueprint. The next critical step is to take that design out of the computer and into the lab to begin real-world testing.
And this all leaves us with a pretty fascinating final thought. As new viral threats inevitably continue to emerge, could these lightning-fast virtual labs become our most powerful first line of defense in the race to find a cure?